Hello everyone, in this video we will practice some NEET previous year questions of biology. This is the part 2 of this session. If you haven't watched my first part, then you will find its link in this top I button. You will also find its link in description as well. So here is your first question. Which of the following is not a method of ex situ conservation? Option 1, in vitro fertilization. Second, national parks. Third, micropropagation. And fourth, cryopreservation. So your answer will be option 2, that is national parks. See, what do we mean by in situ conservation? So in situ conservation means on site conservation. That is when we conserve and protect the whole ecosystem. Its biodiversity at all levels is protected. For example, we can say national parks. National parks are type of in situ conservation. On the other hand, micropropagation, cryopreservation, and in vitro fertilization, that is IVF, these are methods of ex situ conservation. So your correct answer will be national parks, which is in situ conservation. Your second question is identify the correct set of statements. A. The leaflets are modified into pointed hard thorns in citrus and bougainvillea. B. Axillary buds form slender and spirally called tendrils in cucumber and pumpkin. C. Stem is flattened and fleshy in opuntia and modified to perform the function of leaves. D. Rhizophora shows vertically upward growing roots that help to get oxygen for respiration. And E. Subaerially growing stems in grasses and strawberry help in vegetative propagation. Now we have to choose the correct answer from the options which are given below. So see, your answer will be option 4, that is A, B, D and E only. These A, B, D and E are correct. So as we all know that axillary buds of stems get modified into which parts? Axillary buds of stems get modified into woody, straight and pointed thorns. And thorns are found in many plants such as citrus and bougainvillea. So your correct option will be option 4, that is A, B, D and E statements are correct. Let's move on to the next question. Given below are two statements. Statement 1, decomposition is a process in which the detritus is degraded into simpler substances by microbes. And statement 2, decomposition is faster if the detritus is rich in lignin and chitin. In the light of the above statements, choose the correct answer from the options given below. Now see, as we all know that decomposition is a process where detritus is degraded into simpler substances. Or we can say that it is a process by which decomposers break down, decomposers in the sense microorganisms. These microorganisms break down complex organic matter into inorganic substance, right? And the rate of decomposition is controlled by chemical composition of detritus and climatic factors. Your second statement, that it means that statement 1 is correct. Your second statement, that is decomposition is faster if the detritus is rich in lignin and chitin. See, decomposition is slower if detritus is rich in lignin and chitin, and it will be quicker if detritus is rich in nitrogen and water soluble substances like sugars. It means that your statement 2 is incorrect. So your correct answer will be option 3, that is statement 1 is correct but statement 2 is incorrect, because decomposition is faster. When it will be faster? It will be faster when the detritus is rich in nitrogen and water soluble substances. But this process will be slower if the detritus is rich in lignin and chitin. So option 2 is, or we can say statement 2 is your incorrect statement. Fourth question, which one of the following plants shows vexillary aestivation and diadelphous stamens? Option 1, Colchicum autumnale, Pisum sativum, Allium cepa, and Solanum nigrum. So your correct option will be Pisum sativum. See, this vexillary aestivation and diadelphous stamens are characteristic of which family? These are characteristic features of which family? So these are characteristic features of family Fabaceae. Pisum sativum, that is your green pea, it belongs to family Fabaceae. Allium cepa, that is onion, and Colchicum autumnale, that is colchicine, belong to family Liliaceae, and Solanum nigrum belongs to Solanaceae. So your correct option will be option 2, that is Pisum sativum, that is your garden pea, which belongs to family Fabaceae. And this vexillary aestivation and diadelphous stamens are the characteristic feature of which family? That is family Fabaceae. So your correct option will be option 2. Your fifth question is, which one of the following never occurs during mitotic cell division? Your first option, spindle fibers attached to kinetochores of chromosomes. Second, movement of centrioles towards opposite poles. Third, pairing of homologous chromosomes. And fourth, coiling and condensation of the chromatids. So, your correct option will be option 3, that is pairing of homologous chromosomes. So, see, this pairing of homologous chromosome occurs during prophase 1 of meiosis. Coiling and condensation of chromatids, then spindle fiber attachment to the kinetochores, and movement of centrioles towards opposite poles occur in both mitosis and meiosis. So let's move on to the next question. Given below are two statements. Statement 1, the primary CO2 acceptor in C4 plants is phosphoenol pyruvate and is found in the mesophyll cells. And your statement 2 is mesophyll cells of C4 plants lack Rubisco enzyme. In the light of the above statements, choose the correct answer from the options given below. See, as we all know that the primary CO2 acceptor is a 3 carbon molecule, that is phosphoenol pyruvate, that is we say it PEP, and is present in the mesophyll cells. This statement is correct, and mesophyll cells of C4 plants lack Rubisco enzyme. The second statement is also correct, so your answer will be both statement 1 and statement 2 are correct. Question number 7. Which one of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? It involves first, breakdown of proton gradient, second, breakdown of electron gradient, third, movement of protons across the membrane to the stroma, fourth, reduction of NADP and NADPH2 on the stroma side of the membrane. So your correct answer will be breakdown of electron gradient. See, chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthesis. And energy is used to pump protons across a membrane to create a gradient, or we can say a high concentration of protons within the thylakoid lumen. The NADP reductase enzyme is located on the stroma side of the membrane. We all know that this NADP reductase enzyme is located on the stroma side of the membrane. Along with the electrons that come from the acceptor of electrons of PS1, protons are necessary for the reduction of NADP plus to NADPH2, and the process does not involve breaking of electron gradient. So your correct option will be option 2, that is breakdown of electron gradient. Let's move on to the next question. Habitat loss and fragmentation, overexploitation, alien species invasion, and co-extinction are causes for population explosion, competition, biodiversity loss, or natality. So your correct option will be option 3, that is biodiversity loss. C. Habitat loss and fragmentation, overexploitation, alien species invasion, and co-extinction are causes for what? These are causes for biodiversity loss. Your ninth question. What is the net gain of ATP when each molecule of glucose is converted to two molecules of pyruvic acid? Your options are 4, 6, 2, or 8. As we all know, during glycolysis, total 4 ATPs are produced, right? So, total 4 ATPs are produced from one glucose molecule with a net gain of 2 ATPs. So, your correct answer will be option 3, that is 2 ATPs. Let's move on to next question, which is your last question. Girdling experiment was performed by plant physiologists to identify the plant tissue through which water is transported, food is transported. For both water and food transportation, or osmosis is observed. So your correct answer will be option 2, that is food is transported. The Girdling experiment shows that phloem is the tissue responsible for translocation of the food, and that transport takes place in one direction, that is towards the root. So it is the experiment which was performed by plant physiologists to identify the plant tissue through which food is transported.